Thank you, Dieter Beule, for inviting me to contribute to this workshop. My name is Ben Liesfeld from Limbus Medical Technologies, and in my short presentation I will focus on how to validate whole exome diagnostics in order to make it suitable for clinical diagnostics. Let me briefly introduce to you who we are. Limbus is a software development company located in northern Germany. We are a medical device manufacturer, which means that we operate and develop our products according to international medical device standards. Our products are cloud-based and specifically designed to aid in the diagnosis of patients. I'd like to quickly revisit a few definitions to ensure that we have a common understanding of what validation is. The terms verification and validation are often used interchangeably, or at least in a confusing manner. They are, however, the answer to quite different questions. Did we build the product right? Or did we build the right product? You may also insert your assay or diagnostic test in these questions. Verification is the gathering of rather technical evidence that technical requirements have been met. Validation, on the other hand, determines on a higher level if the intended use can be achieved. You may therefore verify that your whole exome pipeline works by simply processing in silico datasets. Validation of a whole exome pipeline, however, requires that you process human DNA in a real-world diagnostic process. You may compare this to the self-driving car. If you ever want to achieve a fully automated process, you must be sure that it works reliably. Without validation, you will never be able to take the hands off the steering wheel. You will have to constantly check and monitor your results, which is just not feasible in the case of whole exomes. If you are performing whole exome sequencing on patients and not just studied subjects, you are required by law to validate your diagnostic process. Last but not least, at least in Germany, genetic tests may only be reimbursed if they have been validated. The steps that we at Limbus follow to onboard new users may serve as a blueprint for anybody who intends to establish an exome diagnostic assay. We start with reviewing and processing data from sequencing runs that have been performed so far. Here, we assess intersample and interrun variability and can predict the outcome of a validation process. Then, we actually perform the validation. The user simply has to provide the data. We will show you how we do that exactly on the next slides. After or during the validation, the users perform an evaluation of usability and features in comparison to existing or other solutions. The important benefit of this three-step process is that the entire assay is ready to be used in clinical routine if the software evaluation is completed successfully. Let's take a closer look at how the validation process should be structured. I'm listing here the differences between the SNV validation and the CNV validation of a whole exome diagnostic assay. Please note that every time I say SNV, I actually mean SNV and indels. I'm not only referring to single nucleotide variants here, but to small variants in contrast to copy number variations. For the SNV validation, there are several reference materials available. These materials have been characterized using multiple analytical methods. Therefore, we are comparing to multiple methods in the validation process. This also means that our target cannot be to achieve 100% sensitivity compared to these multiple methods. Applying a single method, you cannot expect to be as good as multiple reference methods combined. Our target, according to international guidelines, should be something between 95% and 98%, but the guidelines are a few years old, so make sure to be rather on the 98% side than on the lower end. For the CNV validation, unfortunately, no established or suitable reference materials exist. This means that we have to rely on real clinical samples in which variants have been confirmed using methods like ACGH or MLPA. Since we will be comparing to a single reference method, and since our goal is to replace this reference method in clinical routine with exome sequencing, we must achieve 100% sensitivity, or be at least close to that. Let me quickly summarize the most important parameters that you obtain from a diagnostic test validation. Sensitivity tells you the fraction of all true variants in the sample that are detected by your test. Specificity turns this around and tells you how good you are in detecting a negative case. Note that you cannot determine the specificity for SNVs and indels because the number of true negatives cannot be determined. The positive predictive value, or also called precision, indicates the probability of a variant actually being true. The negative predictive value, on the other hand, tells you how sure you can be that a negative result is truly negative. From a diagnostic point of view, a first-line diagnostic test must be as sensitive as possible. We would like to achieve a sensitivity close to 100%. At the same time, we will make a compromise on specificity. However, a low specificity may force us to perform a lot of confirmatory testing. Since reference material is available, the SNV validation process is very much straightforward. The reference samples are processed in the laboratory in the same way as any clinical sample. In the end, the results are compared to the published reference data. Make sure that you use established software tools for this process in such a way that this can be automated. Keep in mind that this process should be repeated every time that the lab process or the software changes. This is the convenience that we provide with varvis. This is an example of the validation results from genome in a bottle samples. Note that you should report the performance characteristics for SNV and indels separately. The guidelines, however, only specify acceptance criteria for all variants together. Geneticists must be aware of the fact that the analytical performance of indel detection is always significantly lower than for SNV detection in hybrid capture-based assays. This is a technical limitation. The CNV validation process is much more complex, on the other hand. It is important to note that the CNV validation should compare every single probe of the reference method to the NGS-based CNV analysis. Here you can see an example of a validation sample with a deletion in LDLR and how the NGS probes are matched to the MLPA probes. The procedure is therefore a true head-to-head comparison with single exon resolution and realistically assesses the performance of the assay. These are the results of the validation process obtained from 40 whole exome samples with CNVs confirmed by MLPA. 40 samples may or may not seem a large number. It is possible, however, that a laboratory does not have to perform a validation from scratch, but merely confirms validation results from the manufacturer, as long as the sequencing parameters are within specifications of the manufacturer. This is the case, for example, for users of varvis. In this case, we make the direct comparison to MLPA in order to replace MLPA with exome sequencing. As you can see, we were able to achieve 100% sensitivity in our cohort of validation samples. The 95% confidence intervals give you an idea of how sure we are to achieve these values. As expected, the analytical performance is much better for deletions than for duplications. We can add validation samples to this pool in the future, which will narrow down the confidence intervals. On the right-hand side, you can see the workflow as it may be implemented in the lab based on this validation. All samples are undergoing the NGS-CNV analysis. A quality control check determines if the CNV analysis can be performed. The negative cases can be determined very quickly, since we confirmed 100% sensitivity. If necessary and if available, confirmatory testing can be performed. If you're interested in getting more hands-on information how exome diagnostics can be performed in practice, I would like to refer you to our satellite meeting, where Halenur Yavuz-Kienle and Rami Abu-Jamra talk about their experience with exome diagnostics and present real-world examples. Thank you.